first of all, I thank an organizer to allowing us to show and give this presentation. So this work was, I'm Masanori Honsho from Kyushu University, and this work is collaboration. This work was done in collaboration with Institute of Rheological Functional Food. So the title is Plasmalogen Supplementation, a promising therapeutic strategy for well-being in an aging population. So, how to, sorry, I have no idea how to.
This is next, this is forward.
Okay. This is forward. Okay, now. Sorry. So let me explain what is the plasmalogen. So plasmalogen is a subclass of glycerol phospholipid, which contains a vinyl ether bond as marked in red dash box here. And the head group is typically ethanolamine and choline, and R1 position and R2 position are occupied by fatty alcohol and fatty acids such as deoxyhexonic acid, respectively. And the plasmalogen is synthesized in cells, and approximately 80% of total phospholipid in human is a plasmalogen. So therefore, the plasmalogen are distributed in the body and play an important role, such as antioxidant and membrane fusion, and so on, like it's shown in blue. But these studies were identified or proposed from using the cells deficient plasmalogen or mice defective in plasmalogen synthesis. But instead, we succeeded to isolate a large amount of plasmalogen from scallops or chickens, and which enable us to test the effect of supplemented plasmalogen in animals, including human. And we found that the plasmalogen has anti-neuroinflammation activity and neurogenesis. So this is showing that the plasmalogen suppresses neuroinflammation in microglia cells. Microglia is primarily immune cell in central nervous system. In this study, we used MG6, a mouse microglia cell line, and we treated the cell with LPS, liposaccharide, which induce neuroinflammation. You can see the elevation of the expression NOS2, pro-inflammatory marker for microglia activation. But when we treated the cell, we cultured the cell in the presence of plasmalogen and followed by treatment with LPS, you can see the reduction of NOS2 expression. So meaning that plasmalogen can suppress the neuroinflammation. And by in vivo, we found that the plasmalogen suppress the accumulation of amyloid beta. So in here, we treated the LPS to control mice and observed the accumulation of A-beta using the A-beta specific antibody here. But when we treated plasmalogen together with LPS, you can see the reduction of accumulation of A-beta. And by administrating plasmalogen in control mice, we could see the enhancement or promotion of neurogenesis in hippocampus, which was observed by the marker of using double colchicine marker for the newly generated neurons. And we also found that the plasmalogen can promote a dendrite formation, as shown here, that we took a primary neuron from mice and cultured this cell in the presence or absence of plasmalogen for 6 and 40 days. You can see the highly branched dendrite formation in here. So this, I just summarized my first part. So plasmalogen inhibits neuroinflammation and the amyloid beta accumulation in the brain, suggesting that its preventive effect against Alzheimer's disease. And the second thing is that the plasmalogen promotes neurogenesis in normal mice and the senescent accelerated mice, although I haven't showed the data of senescent accelerated mice due to the time limitation. But this shows that the plasmalogen has potential to be an effective treatment for Alzheimer's disease. So based on these in vitro and in vivo study, we performed a clinical trial in patients with mild cognitive impairment, MCI, and mild Alzheimer's disease under the multicenter and randomized double-blind placebo-controlled trial. So, and here that we recruited 178 MCI patients and 98 mild AD patients recruited and enrolled, and these participants received purified plasmalogen 1 milligram per day for 24 weeks or placebo. And this is a result of the effect of plasmalogen on cognitive function in MCI, assessed by MMSE. So y-axis shows the difference from the score of MMSE from the starting point to 24 weeks. And here we focus on the orientation to time, because the MMSE has several tasks, but here we focus on orientation time, which is one of the earliest events affected. And also we focus on orientation to place, which becomes impaired as the disease progress. So in left panel, in placebo group, you can see the decline or decrease of MMSE score, but in case of plasmalogen treated or intake human group, you can see the decline of the score. So although we cannot see the significant difference between these one, but in right panel, you can see the increase of improvement of MMSE score compared to the placebo group with a significant difference. So, and this is a result of the effect of plasmalogen in cognitive function in mild AD. Here we use the vascular test, which is a comprehensive memory assessment tool. So here we performed the sub-analysis by gender and age. So here we found in plasmalogen group, we can see the improvement vascular score in plasmalogen group compared to the placebo group with significantly. And here this shows that this graph shows the age differences. So we found that the 77 or younger person shows the improvement of vascular score compared to the placebo group. And we also performed the clinical test against the moderate or severe Alzheimer's disease patient under the multi-center and open label trial without instead of randomized control trial, because placebo effect is known to be almost absent in disease advantage stage in Alzheimer's disease. So here that we recruit 57 moderate Alzheimer's disease and severe Alzheimer's disease 18, and the participant received 1 milligram plasmalogen per day for 3 months. So this is a result. So here the summarized result, so showing the improvement in moderate or severe AD groups. And the blue shows that the improvement of MME score 2.0 or more, and the white bar shows that no change, MME score plus minus 1, and the gray bar show worsening 2.0 or more. So by, as you can see here, that the blue bar, so over the 50% of moderate patients or approximately 30% of severe AD patients show the improvement of MMEC score. So these results indicate that the plasmalogen have function to improve the cognition. So in addition to the effect of plasmalogen, we found that plasmalogen enhanced the innate immune system. So actually the plasmalogen activated natural killer cell through activation of GPCR and enhance the synthesis of perforin, which can make a pore in the target cells. The plasmalogen mediated the activation of natural killer cells can suppress virus replication. Here that we use the control mice and under the normal condition or plasmalogen treated condition, and after that we infected the virus and then tested the remaining of virus or replicated the virus amount in these tissues. Clearly we can see the reduction of the virus amount by the plasmalogen treatment. Okay, so, and finally that we found that the plasmalogen can promote the muscle growth. As you can see, the increased number of diameter of muscle shown in here. Although we are not sure about the, we are still trying to identify the molecular mechanism, but we found that after the plasmalogen treatment, we could see the elevation of MyoD expression here, as shown here, and this MyoD is highly expressed. It is known that the MyoD is highly expressed in fast twitch, and this MyoD is important or critical transcription factor in muscle differentiation. So the highly expression that the fast twitch means this is very important because the decrease of fast twitch fiber is directly contributed to the muscle weakness or physical dysfunction in sarcopenia patient. So I think we hope that plasmalogen can suppress the sarcopenia. So this is the last slide, the summarized slide. So I showed that the plasmalogen enhance the cognition and suppress the neuroinflammation and activate the neuroinflammation and promote the muscle generation. So we think that this effect of plasmalogen collectively support the health span and extending the healthy lifespan. So thank you for your attention.
Thank you, Professor Masanori. Any questions? Yeah, please. Please. No?
I don't think it's been looked at for survival in mice, has it? It would be interesting to do a longevity study on this and see if the mice live longer. And the other thought I had is related to a muscular dystrophy model might be interesting for the muscle phenotype.
Okay, I don't see any other questions. Thanks again. And